In 2021, a miracle drug successfully turned off the exact mutation causing a patient's cancer. And the scans, they came back totally clear. The targeted therapy worked flawlessly. But then 6 months later, the cancer was back. And this time, it was completely immune to the drug.
Yeah, which is just, it's devastating. And it happens far too often.
Exactly. And I know you, our listener, are here today because you really want to understand the mechanics behind stories like this. You want to know what actually happens at the cellular level when biology basically outsmarts our best medicine.
Right.
So today we are unpacking a really groundbreaking February 2026 paper in Nature Cancer that finally explains how identical cancer cells literally, rewire their own machinery to survive. We're doing a deep dive into a revolutionary platform called GENEVA. And the core problem we're tackling here is a massive bottleneck in how we test drugs and decode tumor resistance.
Yeah, and if we connect this to the bigger picture, that bottleneck is entirely about scale and environment.
Right.
Because for decades, testing a cancer drug before human trials meant relying on mouse xenografts.
Right, the standard model.
Exactly. You take one specific human tumor line and graft it into a mouse and apply the drug. But it's a really slow, linear process. You're testing one single genetic profile against one drug at a time. And given the infinite genetic diversity of cancer, even within a single patient, this one-to-one modeling is fundamentally incapable of predicting how a really complex, mutating disease will react across a diverse population.
Which brings us to the paradigm shift, GENEVA, which stands for Genetically Diverse and Endogenously Controlled Phenotypic Variation Assay. It's basically a way to test dozens of unique tumor genetic profiles simultaneously in a single experiment inside a single living mouse.
It's wild. It accelerates high-throughput drug screening by an order of magnitude. We're moving away from that linear testing and into true multidimensional analysis.
Okay, let's unpack this, because I understand the difference between standard CDX, cell line-derived xenografts, and PDX, the patient-derived xenografts.
Yeah.
You're just taking cells grown in a lab versus cells taken straight from a patient.
Right, exactly.
But taking dozens of these lines, maybe, 50 different cancer cell lines, mixing them all together, and injecting that single mixed pool into one mouse to create what they call a tumor mosaic. I have to push back here. Doesn't mixing 50 different tumor types into one mouse create a completely unnatural chemical environment?
Yeah.
Because they're competing for resources, right? They're signaling to each other, altering the microenvironment. Doesn't that just skew the data compared to a real human tumor?
That is the exact reason GENEVA is designed as an endogenously controlled assay. Because think about it, when you put 50 different cell lines into 50 different mice, the biological variation between those 50 mice is huge.
Oh, like different immune responses?
Exactly. Differences in immune response, hormone levels, drug absorption rates, all of that creates massive statistical noise. But by putting the mosaic into a single mouse, every single cell line is subjected to the exact same drug concentration, the exact same host metabolism, and the exact same microenvironment.
Ah, I see. So the environmental variables are perfectly controlled.
Right. Any difference in survival is definitively linked to the cell's internal genetic profile, not just a quirk of the host mouse.
Okay, testing drugs the old way is essentially like testing one car on one track at a time to see if it crashes.
Yeah, good analogy.
But GENEVA is like putting 50 different cars on the track at once to see who survives the demolition derby.
Exactly. That's exactly what it is.
But practically speaking, how do you read those results? You hit this mosaic tumor with this demolition derby of targeted therapies, and at the end of the experiment, you have a mass of surviving cells. But a surviving cancer cell from line A looks just like a surviving cancer cell from line B. So how do you possibly separate and identify who survived?
Well, you give them a molecular barcode before the experiment even begins.
Yeah.
This is done through a process called hashing, and they utilize lipid oligonucleotides, or total CK antibodies.
Okay, wait, let's break down the mechanics of that. How does a lipid oligonucleotide actually attach to a cell without destroying it?
It's actually really elegant. It exploits the natural structure of the cell itself. So the cell membrane is a lipid bilayer, right? It's made of fat. The hashing molecule has a highly lipophilic end, basically a fat-loving tail, that naturally inserts and anchors itself right into the cancer cell's membrane.
Oh, so it just slides right in.
Exactly. And attached to that anchor is the oligonucleotide, which is just a short synthetic strand of nucleotides. It acts as a unique sequence, a literal barcode.
What about the other one, total CK?
Right. Alternatively, they use total CK, which relies on antibodies that bind to ubiquitous surface proteins found on all human cells. And they drag along a specific poly-A tail barcode. Either way, before mixing, cell line A gets barcode 1, cell line B gets barcode 2.
So they basically go into the mouse wearing these barcode backpacks.
Yeah, barcode backpacks. I like that.
So the tumor grows, you apply the drug, and then you harvest whatever survives. But what actually reads the backpacks?
That would be single-cell RNA sequencing, or cRNA-seq. They extract the surviving mosaic tumor and use microfluidics to separate it back down. So every individual cell is encapsulated in its own microscopic water and oil droplet.
Wait, it literally traps one single cell per droplet.
Just one single cell per droplet. And inside that droplet, the cell is lysed. It's broken open. The sequencer then captures the cell's entire natural messenger RNA, its transcriptome, alongside that synthetic barcode we added earlier.
Okay, let's do a quick recap here to make sure we're all tracking. So, bullet 1. Xenografts are vital for cancer research, but they're traditionally way too slow because they test one variable at a time. Bullet 2. GENEVA obliterates that bottleneck by pooling dozens of lines into one mosaic tumor inside one mouse. And bullet 3. The absolute genius here is that by combining lipid-anchored barcodes with single-cell RNA sequencing, you don't just see which genetic profile survived the drug, you see exactly what genes that specific cell is expressing at the moment of survival, which is incredible.
It completely changes the speed of discovery. You're no longer guessing why a drug failed across a population. You are reading the exact survival manual of every variant simultaneously.
So I want you, our listener, to think about your own problem solving at work for a second. How often are you testing variables one by one when you could be running a mosaic test to see all the outcomes at once? Keep that in mind as we look at GENEVA's first big target.
Which was a pilot study, right?
Yeah. To prove this platform wasn't just theoretical, they ran a pilot study on known biological quantities. They pooled 11 different cell lines. And within this pool was the A375 line, which carries a specific mutation called BRAFV600E, and the MIA-PACA2 line, which carries the KRAS G12C mutation.
Okay, two very notorious cancer targets. Exactly. And then they applied vemurafenib, which is a targeted inhibitor for the BRAF mutation, and ARS-1620, an inhibitor for KRAS G12C.
Right, so you dose the mosaic. And the vemurafenib selectively annihilates the A375 cells, while the ARS-1620 wipes out the MIA-PACA2 cells. It confirms the baseline. But here's where it gets really interesting, because as we established, single-cell RNA sequencing doesn't just give you a body count, it gives you the psychological profile of the survivors.
It reveals intrapopulation heterogeneity.
That's a mouthful. What does that mean in practice?
Basically, when researchers isolated the MIA-PACA2 cells that managed to survive the KRAS inhibitor, they analyzed their gene expression landscape. And the RNA profile of these surviving cells deviated from random chance with a massive Z-score of 6.65.
A Z-score of 6.65, that's statistically huge. That implies this wasn't an accident.
Exactly. In statistical terms, that implies a highly coordinated, non-random biological adaptation.
So they didn't just survive because they were lucky or, because the drug somehow didn't reach them in the tumor. The surviving cells of the exact same genetic line had fundamentally altered their internal machinery.
The machinery was entirely rewired. It shows that even genetically identical cells living in the exact same environment react differently to the same stressor. Some just find an alternate pathway to survive.
So to summarize the subtopic, GENEVA successfully mapped the vulnerabilities of specific mutations like BRAF and KRA, but more importantly, it proved intrapopulation heterogeneity, meaning targeted drugs force identical cells into diverse, highly specific survival states.
Right. It really forces you to think like the cancer cell.
Yeah, and listener, I want to pose a question to you from our expert. If you were a cancer cell trying to survive a targeted bomb, how would you change your internal machinery to stay alive? Because that leads us right into the next major discovery.
Yeah, if your primary growth pathway is suddenly bombed, how do you keep the lights on? You find a secondary power source. And in the case of KRAS G12C, that power source was the mitochondria.
KRAS G12C is the villain in so many of these cases. I mean, it's a signaling protein that acts like a cellular on-off switch for growth. And in these tumors, it is literally jammed in the on position. In recent years, drugs like sotorasib finally managed to block it. But as we established at the start of the deep dive, the cancer often roars back within 6 months.
Right. And we didn't fully know why. But when the researchers used GENEVA on a pool of 8 different KRAS G12C lines treated with the inhibitor ARS-1620, the cRNA-seq flagged a real paradox. The surviving cells consistently showed significantly fewer mitochondrial transcripts compared to the untreated cells.
Wait. If they have fewer mitochondrial transcripts, does that mean they're powering down? Like, are they shrinking their energy footprint to essentially hibernate through the chemical attack?
See, that was the initial assumption.
Yeah.
It makes sense, right?
Uh-huh.
But when they measured the oxygen consumption rate, the OCR, they found the exact opposite. Yeah. They took a line of cells called H2030 and continuously exposed them to the drug for 34 days, creating a hardened persister line. When they hit these persisters with an acute dose of sotatercept, the cells didn't power down at all. Their OCR and their production of ATP, the raw energy currency of the cell, rapidly spiked.
How is that mechanically possible, though, if their main growth pathway is blocked and their mitochondrial gene expression is supposedly way down? Where is this massive energy spike coming from?
Okay, so blocking KRAS essentially starves the cell of its preferred fuel process, which is glycolysis. That's basically fermenting glucose outside the mitochondria. To avoid starving to death, the cell is forced into a compensatory surge. It drastically ramps up oxidative phosphorylation inside the mitochondria.
Oh, wow.
It is a desperate, highly efficient energy burn. And the cells with lower baseline mitochondrial transcripts were somehow uniquely primed to survive this shift without burning themselves out from oxidative stress. So the drug was inadvertently triggering an alternative survival engine.
That is wild. So you block the cancer's main engine, and the resulting starvation forces it to hit a hidden nitrous button in the mitochondria. But if that metabolic pivot is the escape route, the next logical step is to close it, right?
Which is exactly how you build a graded evidence combinatorial therapy. If the KRAS inhibitor forces the cell to rely entirely on its mitochondria, you hit the mitochondria at the exact same time. So researchers paired the KRAS inhibitor with a drug called antimycin.
Okay, antimycin is a mitochondrial complex 3 inhibitor, correct. Let's clarify how that actually shuts down the engine for the listener.
Sure. So in the inner membrane of the mitochondria, you have the electron transport chain. It passes electrons down a line of protein complexes to generate ATP. Complex 3 is a crucial middleman in that chain. Antimycin binds to it and permanently halts the flow of electrons. The proton gradient just collapses and ATP production flatlines.
So when they combine the KRAS inhibitor to block glycolysis and antimycin to break the electron transport chain?
The result was a complete synergistic wipeout. The persister cells had no metabolic fallback left, and they died.
Okay, let's do our 5-minute recap to lock down this mechanism. Bullet 1. Cancer cells that survive a targeted KRAS blockade don't just sit there, they show altered mitochondrial states. Bullet 2. The drug actually triggers a desperate temporary spike in mitochondrial energy production. And bullet 3, blocking this energy spike with antimycin restores the cancer drug's lethal power.
Right, exactly.
It's like discovering the cancer escapes through a basement window and immediately boarding it up. But listener, what happens when the cancer doesn't just find a new window? What happens when it fundamentally changes its physical shape to slip through the cracks? Because that is exactly what GENEVA revealed when they took this out of the Petri dish and into living mammals.
Yeah, and what's fascinating here is the stark contrast between in vitro and in vivo environments. A plastic Petri dish doesn't enforce physical constraints. It doesn't have an extracellular matrix or stromal cells or a circulatory system. When GENEVA mapped these tumors inside living xenografts, entirely new resistance pathways emerged that were completely invisible in the plastic dish.
And the first major in vivo escape route GENEVA flagged was the mTOR pathway.
Right. The mTOR pathway regulates cell growth and protein synthesis. The living microenvironment pushed the surviving cancer cells to heavily upregulate mTOR, signaling to bypass the KRAS blockade. And when researchers paired the KRAS inhibitor with INK128, which is an mTOR inhibitor, they saw a massive synergistic collapse in tumor volume inside the mice.
But the second pathway they found in vivo is where the biology gets genuinely architectural. It's called EMT, or epithelial-to-mesenchymal transition.
This is a really crucial concept.
Let's use an analogy for this. Imagine the cancer cells in their standard epithelial state are like a Roman phalanx, you know? They're rigidly locked together, shield to shield, held tightly by proteins like E-cadherin. This rigid, tightly packed formation is what creates the solid mass of a tumor.
And the targeted drug acts like an artillery strike on that phalanx.
Exactly. But the survivors don't just stand there and take it. They undergo EMT, they drop their shields, they break those tight intercellular bonds and transform into mesenchymal cells. These are solitary, highly mobile fluid units. They can physically slide away from the primary tumor, navigate through the extracellular matrix, and evade the localized concentration of the drug.
EMT completely remodels the cell's cytoskeleton. I mean, it is a classic mechanism for cancer metastasis. It's how a tumor spreads to the lungs or the brain. But Geneva proved that EMT is also a direct, immediate survival reflex against KRAS inhibitors in a living environment.
So they literally changed their uniforms to escape.
Exactly. So to counteract this, researchers needed to stop the cells from dropping their shields, and they introduced galunisertib.
Break down the mechanism of galunisertib for us. How does it freeze the phalanx?
Galunisertib is a TGF-beta receptor inhibitor. TGF-beta is a cytokine, a signaling protein in the microenvironment that essentially broadcasts the order to undergo EMT. Galunisertib binds to the receptors on the cancer cells, basically deafening them to that signal. When researchers combined the KRAS inhibitor with galunisertib, the cells were forced to remain in their rigid epithelial state, and tumor growth was dramatically stunted.
That's brilliant. But correlation is not causation. Observing a gene's activation during survival doesn't inherently prove that the gene caused the survival. How do they prove beyond a shadow of a doubt that these specific pathways, mTOR, EMT, mitochondrial metabolism, were the literal drivers of resistance?
They validated Geneva's findings using an in vivo CRISPR screen. This is essentially the ultimate algorithm of biological truth.
Okay, walk us through how CRISPR interference functions differently than standard CRISPR-Cas9, because it's not just snipping DNA, right?
No, it doesn't cut the DNA at all. They use a system called dual-guide dCas9-KRB. The D stands for dead. The Cas9 protein has been mutated, so its nucleus or cutting ability is deactivated. It still uses guide RNAs to find a highly specific gene promoter, but instead of cutting, it just parks there.
Oh, it just sits on the DNA.
Yep, and attached to this dead Cas9 is a KAB domain. KAB recruits chromatin and remodeling complexes that tightly wind up the DNA around that specific gene, physically blocking transcription. It basically acts as a highly precise dimmer switch, turning specific genes off without altering the genetic code itself.
So they took the living mosaic tumors and systematically used this dimmer switch to turn off the exact genes Geneva had flagged. They essentially ran a live-action simulation. Like, if we manually switch off the mTOR pathway or the EMT effectors, do the cells die faster?
And the results match perfectly. When they turned down the mTOR and EMT effector genes, the cells became highly sensitized to the KRAS drug. They lost their escape routes and died rapidly. Conversely, when they used CRISPR to turn down genes responsible for mitochondrial ribosomes, essentially enforcing the metabolic state the cancer was trying to achieve, the cells were profoundly protected.
It's just staggering. Okay, let's recap this section. Bullet 1, Geneva identified new drug targets like the mTOR and EMT pathways that only show up in living environments. Bullet 2, EMT allows cells to physically morph and evade treatments in vivo. Bullet 3, CRISPR genetic editing validated that hitting these secondary pathways creates powerful combination therapies.
Right. And listener, I have a question for you based on all this. If tumor resistance is this dynamic, how should we be designing clinical trials for human patients tomorrow?
That's the million-dollar question.
It dictates that monotherapy, giving a patient a single targeted drug and waiting to see if it works is fundamentally outdated. We cannot wait for the tumor to evolve and then try to guess its new form.
So what does this all mean? We have navigated an immense density of information today. We started with the unscalable bottleneck of traditional mouse models. We explored the Geneva platform, throwing dozens of distinct tumor profiles into a single, perfectly controlled endogenous environment.
Yeah.
We examined the mechanics of hashing with lipid oligonucleotides and cRNA-seq, allowing us to read the exact biological state of every single surviving cell. And the data showed us that cancer survives targeted attacks by triggering massive mitochondrial energy spikes and undergoing radical structural remodeling through EMT.
And for you listening, whether you are looking at medical investments, caring for a loved one, or just fascinated by biology, Geneva represents a massive acceleration in how fast we can find out why a drug fails and exactly what second drug to pair it with to succeed.
Absolutely. And I want to leave you with a final provocative thought. Throughout this deep dive, we've discussed using Geneva to pool established cell lines to map general resistance. But if this platform can simultaneously test 50 unique genetic profiles inside a single controlled environment, could we eventually create a digital twin of this platform?
Now, that's an interesting idea.
Right. What if, in the near future, we don't just pull generic cancer lines, but we take a biopsy of your specific mutating tumor, break it down into its different subpopulations, hash the cells, and run a personalized Geneva assay? We could predict your exact resistance mechanism before you even take your first pill.
It elevates oncology from a reactive science of statistical guessing to a truly predictive, personalized discipline.
Thank you for taking this incredibly deep, rigorous dive with us into the source material today. You are the reason we push past the surface level summaries. Next time you think about medical precision, remember that biology is never static. It is a constantly shifting, adapting mosaic. But finally, we are building the tools to map it.